Good evening, students. Welcome to Sankalp Learning Classes. Today we are going to start the new chapter, that is Microbes in Human Welfare. So this is the 8th chapter from the biology. So already we have completed the 7th chapter, that is Human Health and Diseases. So this is the next chapter, so that is the Microbes in Human Welfare. Here, the first thing is, what is the meaning of microbes, right? So microbes means, you know, that microorganisms. Microorganisms, micro means which are not able to see and with the naked eye. So we need microscope or high level compound microscopes or high advanced electron microscopes to observe these microorganisms. Then what is the human welfare means? How they are going to help in a different purpose of for human beings, in a different way they are going to help the human beings. So commonly, what do you think? Microbes means they cause the disease, so that will come to our mind. But the thing is, microbes, they are helping in many purpose. Okay, they are helping in many functions in our day to day life. So that thing we are studying in this chapter, not about the diseases, but how they are going to help us in our day-to-day life functions. Okay, that we are going to study in this chapter. Then, where they are present, microbes commonly they are present everywhere. So they are not seen by our naked eyes. So in soil, water, inside our bodies, on our bodies, and animals, plants, everywhere they are present. Okay, they are present in a different sites also. For example, high thermal, in high temperature also, and deep soil, and where the layer of soil is thick. So in that condition also they are present. Okay, so in acidic environment, in different environmental conditions, we can see the microorganisms. Okay, then organisms that are visible only under microscope are known as microbes. They are present everywhere, almost in the different habitat, that is deep ocean, in plants, animals, everywhere they are found. The common microbes, what we know, that is the common microbes, example for microbes, they are viruses, viroids, prions, bacteria, fungi, and protozoans, and some algae. So these all are the some examples for the microbes. Okay, microbes are harmful to humans and also useful to mankind in many ways. Yes, as I said, they cause the diseases, and they are harmful for the human beings, but they are also helpful for the human beings in many ways. Okay, how they are going to help the human beings in different functions, in different fields. So in household products and sewage treatment, and the second one in industry for the making of medicines, and biogas production, and biocontrol agent, and the formation and production of the biofertilizers. So in these all fields, the microbes are very helpful for the human beings. So let us see one by one in detail. Okay, the first one, microbes in household products. Household products means what we use in our home, in our day-to-day life. Okay. For example, so here many examples how these microbes are going to help in our household product, to make the household products. Here, lactic acid bacteria, that is lab bacteria, so that is the lactobacillus, also called as lactobacillus, so which helps for the formation of the curd. Okay, so curd formation from the milk. You know that ಕಾಮನ್ ಆಗಿ ನೀವು ನೋಡಿರ್ತೀರ, ನೀವು ಹಾಲನ್ನ ಹೆಪ್ಪಾಕಿದಾಗ ಏನಾಗತ್ತೆ, ಅದು ಬೆಳಿಗ್ಗೆ ಆಗೋದ್ರೊಳಗೆ ಮೊಸರಾಗಿರತ್ತೆ ಅಲ್ವಾ. ಹಾಲಿಗೆ ಸ್ವಲ್ಪ ಮೊಸರನ್ನ ಹಾಕಿದಾಗ ಏನಾಗತ್ತೆ, ಅದು ಹೆಪ್ಪ ಆಗಿ ಏನಾಗಿರುತ್ತೆ, ಮೊಸರಾಗಿರುತ್ತೆ. So how this process will take place? So that is because of the bacteria. Which bacteria? That is lactic acid bacteria or lactobacillus. ಈ lactobacillus ಏನ್ ಮಾಡುತ್ತೆ? That coagulates the and digests the milk protein and it convert it into a curd. Okay, so lactobacillus ಏನ್ ಮಾಡುತ್ತೆ? Milk in the curd formation ಗೆ help ಮಾಡುತ್ತೆ. Just imagine, so if there is no bacteria, then how milk will convert into curd? So there is no other ways, right? And improves the nutritional quality of curd by increasing the vitamin B12, so which add the some nutrients also, that is vitamin B12, and check the disease causing microbes in the stomach. So the first thing is, that is formation of the curd. Okay, which bacteria is helping, that is very important. And the second most important thing is formation for the production of the cheese. Okay, so cheese in our day to day life we use in a different products to make the different products, right? Here Swiss cheese, that is produced by the bacteria that is called as Propionibacterium shermani. Propionibacterium shermani. Okay, then large holes in Swiss cheese is due to the uh, release of the carbon dioxide, that is by the fermentation process. So cheese, what you see, that is formed by the fermentation process of this bacterium. And one more important type of the cheese, that is Roquefort cheese. Penicillium roqueforti, that is the bacteria responsible for the uh, formation of this cheese. Yes, this is the Roquefort cheese. This cheese is called Roquefort cheese. Okay, so this cheese that is fermented by the bacteria Penicillium roqueforti. Okay, so it gives the particular flavor to the cheese. Okay, for the formation of cheese, it is very important. These two bacteria's name are very important. Then toddy. So what is this toddy? Toddy is the fermentation of sap from the palms. So this is the drink that is prepared from the palm plant. Yes, this is the palm plant and this is the toddy. Okay, so that is fermentation of this toddy, that is by the bacteria. Okay, so that is the drink that is obtained from the palm plant. So there is a fermentation process is required, so that will be by the microbes. Okay, then one more thing is idli dosa, so batter, that is fermented by the bacteria, that is mainly different types of the bacteria. ಇಡ್ಲಿ ದೋಸಾ ಬ್ಯಾಟರ್, ಸೊ ನೀವು ಕಾಮನ್ ಆಗಿ ನೋಡಿರ್ತೀರ ಅಲ್ವಾ, ಇಡ್ಲಿ ಮತ್ತೆ ದೋಸಾನ ಮಾಡ್ಬೇಕಾದ್ರೆ ಏನ್ ಮಾಡಿರ್ತಾರೆ, ಅದನ್ನ ರುಬ್ಬಿಡ್ತಾರೆ ಅಲ್ವಾ. ಸೊ ರುಬ್ಬಿಟ್ಟಾಗ ಏನಾಗತ್ತೆ, ಅಲ್ಲಿ ಫರ್ಮೆಂಟೇಶನ್ ಆಗಿ ಅದನ್ನ ನಾವು ದೋಸೆ ಅಥವಾ ಇಡ್ಲಿ ಮಾಡಿದಾಗ ಅಲ್ಲಿ ತುಂಬಾ ಮೃದುವಾದಂಥ ಒಂದು ದೋಸೆ ಅಥವಾ ಇಡ್ಲಿ ಆಗಕ್ಕೆ ಅದು ಹೆಲ್ಪ್ ಆಗತ್ತೆ ಅಲ್ವಾ. ಈ ಮೃದುವಾದಂಥ ಇಡ್ಲಿ ಮತ್ತೆ ದೋಸೆ ಉಂಟಾಗಕ್ಕೆ ಕಾರಣ ಏನು, ಅಲ್ಲಿ fermentation process agide. The bacteria is going to help that process. Okay, and formation of the bread. ಬ್ರೆಡ್ ಫಾರ್ಮೇಶನ್ ಮಾಡಲಿಕ್ಕೆ ನೀವು ಕಾಮನ್ ಆಗಿ ನೋಡಿದ್ದೀರಾ. ಸೊ ಯು ಸಿ ದ ಬ್ರೆಡ್ ಇನ್ ಬೇಕರೀಸ್, ದಟ್ ಈಸ್ ವೆರಿ ಸ್ಮೂತ್ ಅಂಡ್ ವಿಚ್ ಹ್ಯಾಸ್ ದ ಹೋಲ್ಸ್, ಸ್ಮಾಲ್ ಹೋಲ್ಸ್ ಅಂಡ್ ಸ್ಮೂತ್, ರೈಟ್? ಸೊ ದಟ್ ಈಸ್ ಬಿಕಾಸ್ ಆಫ್ ದಿ that is yeast, that is called as Saccharomyces cerevisiae. Okay, that yeast is known as Saccharomyces cerevisiae. Okay, so these all are the microorganisms which are going to help in the formation of curd, cheese, toddy, and the dough. Okay, so here you should remember which of the bacteria or fungi. Here yeast is the fungi. Okay, so they are going to help in the formation of these all household products. Now, just imagine if there is no bacteria, or there is no these type of the microbes which help in these all process, so we can't imagine our life, right? So there are many processes like this that is completed by the microbes. Then microbes in industrial products. So industrial products like antibiotics and uh, some alcoholic products, bioactive molecules, chemicals, and enzymes. So these all are the industrial products that is helped by the that will takes place with the help of the microbes. For example, bioactive molecules. So in detail we are studying in the next PPT. Okay, so cyclosporin A and the statin, and the production of some chemicals like acids, citric acid, acetic acid, lactic acid. So different types of the acids they are produced by the uh, microbes. Okay, then some enzymes like lipase, pectinase and protease, streptokinase, renate and alpha amylase, these all are the enzymes that is produced by the some microorganisms. Okay, so let us start with the first one, fermented beverages. Fermented beverages means like wine, beer and whiskey. So beverages like wine, brandy, whiskey. So these all are the fermented beverages. So these fermented beverages need to be made on large scale, and thus large vessels are used, that is called as the fermenters or bioreactors. Okay, so here large number of microbes are used to make the beverages, different type of beverages like vodka, whiskey, rum, wines and brandy. So these all are the beverages where the microorganisms are used. Okay, and there is large fermenters are needed, so that is called as bioreactors. Raw materials are needed for that. For example, for the wine preparation, grapes are needed and some fruits are needed, right? So fermentation of that raw materials with the help of some microbes like yeast, so saccharomyces, that is also called as brewer's yeast, because which is used in the beverage formation, beverage making, right? And some saccharomyces that is used to make the wine and other beverages. Okay, then alcoholic drinks are produced with the distillation of whiskey, rum and brandy, and without distillation, wine and beer. Here alcoholic drinks, they are produced by the distillation process of this whiskey, rum and the brandy, and without a distillation process we get the wine and beer, that is mainly made up of the fruits. Okay, so this is the first thing where the microbes are used in the industrial purpose, industrial area. That is the first one, beverages, making of beverages. So that is very important. Okay, here, some raw materials are used. For example, rice is used to make the beverage, and apple is also used, and potato is also used to make the vodka, and some cereals are used to make the whiskey, and some fruits are like grapes are used to make the wines. So these all are very important raw materials. When the raw materials they are treated with the some microorganisms like yeast species, then they'll form the alcoholic products. Okay, that is the use of microbes in beverages. Okay, to make the beverages and to form the organic acids. The second important thing is to make the organic acids. Here, citric acid that is produced by the Aspergillus niger, that is the fungus. Then acetic acid, that is produced by the Acetobacter aceti, very important. Then butyric acid, Clostridium butylicum, so that is also bacteria. Lactic acid, that is Lactobacillus. And fumaric acid, Rhizopus nigricans, that is fungi. And gluconic acid, Aspergillus penicillium and the mucor. Oxalic acid, that is produced by the Aspergillus niger. Okay, so these all are the acids which are produced by the microbes. So these all are the microbes that you should remember. Okay, so these microbes are helping to produce the some organic acids also. Then antibiotics. So what is the meaning of antibiotic? So anti means against, bio means biotic, or bio means life. Against life. So then what is that? So this antibiotic term was coined by Salman Waxman. So here the penicillin was the first antibiotic to be discovered by Alexander Fleming in Staphylococcus. Here, the first antibiotic, that is the penicillin, that is derived, that is obtained from the Penicillium notatum, and the scientist's name is Alexander Fleming. Okay, he was working with the bacteria Staphylococcus, and he grew bacteria Staphylococcus in petri dish. After few days, he saw that a mold had developed and contaminated the culture of the Staphylococcus. He examined that mold and found that the mold culture prevented the growth of the Streptococcus. So, Illy, the some chemicals that is obtained from the living organisms, living microbes, that is used to control the growth of the another microbe, that is called as the antibiotic. Okay, so ಇಲ್ಲಿ ಅಲೆಕ್ಸಾಂಡರ್ ಫ್ಲೆಮಿಂಗ್ ಒಂದು ಎಕ್ಸ್ಪೆರಿಮೆಂಟ್ ಮಾಡಬೇಕಾದ್ರೆ ಏನಾಯ್ತು, some molds means that is fungus. Okay, some fungus, what he observed, some fungus that is controlling the growth of the bacteria. Okay, he was working on the Staphylococcus bacteria, and some molds, some fungi that is controlling the growth of the that bacteria. And he observed that that fungi has the some capacity to have some what you say, some chemicals to control that growth of the bacteria. So he observed that, and he extracted that chemical that is known as the penicillin from the penicillium. Okay, so from the Penicillium notatum mold. Okay, here, the penicillium as an effective antibiotic was established later by Ernest Stein on the Harvard floor. So these all are the extra things for your information. So like this, these antibiotics are discovered. Okay, so antibiotics means which are obtained from the microbes, and they are controlling the growth of the some other microorganisms. Okay, living microorganisms. Then next one is production of enzymes. Yes, this is about the antibiotics. Then production of the enzymes. Here lipase, that is the enzyme. So this lipase is, that is Candida lipolytica, so that is the microbe name. So this lipase present in the detergent helpful in removing the oily stains from the clothes. So these are the enzymes present in the detergent that helps to remove the oily stains. Pectinase and protease, so that are used to clear the fruit juices during the bottling. Thus homemade juices are pulpy while the bottled juices are clear. So here, why the bottled juices are clear? That is because of the pectinase and protease. Okay, and one more very important, streptokinase, that is obtained from the streptococcus, that is used as a clot buster for removing the blood clots from the blood vessels of the patients who have suffered from the myocardial infection. Here ಬ್ಲಡ್ ಕ್ಲಾಟ್ಸ್, ಸೊ ವಿದಿನ್ ದ ಬ್ಲಡ್ ವೆಸಲ್ಸ್, ಈಗ ಬ್ಲಡ್ ವೆಸಲ್ಸ್ ಅಲ್ಲಿ ಬ್ಲಡ್ ಕ್ಲಾಟ್ ಆಗಿದೆ ಅಂದ್ರೆ ಆ ಬ್ಲಡ್ ಕ್ಲಾಟ್ ಅನ್ನ ರಿಮೂವ್ ಮಾಡಲಿಕ್ಕೆ ವಿ ಯೂಸ್ ದ ಎನ್ಸೈಮ್ ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ದ ಸ್ಟ್ರೆಪ್ಟೋಕೈನೇಸ್. ಓಕೆ, then rennet and alpha amylase, that is also used here. Rennet, that is used in the manufacturing of cheese, and alpha amylase, that is used for the digestion of starch, softening of the bread, that for that purpose. Okay, so these are the some enzymes, and these are the some functions, and how they are used in a different purpose, so that we studied, and these all are obtained from the microbes. Then bioactive molecules. So there are two bioactive molecules examples are given in your syllabus. The first one is cyclosporin A. Cyclosporin A, that is produced by the fungus Trichoderma polysporum. It is used as an immunosuppressive agent during organ transplant. So organ transplant ಮಾಡಿದಾಗ ಏನಾಗುತ್ತೆ, our body cells they detect as a new organ as a foreign particle, foreign one. So they start to attack on that. So it causes some damage to our body. So immunosuppressants means they suppress the immune activity, the production of antibodies in our body. So that is the cyclosporin A. Then second one, statin, produced by a type of yeast that is Monascus purpureus. That statin is produced by this Monascus purpureus. It is used for lowering the levels of blood cholesterol as it inhibits the enzyme involved in the cholesterol synthesis. So it is used to lower the blood cholesterol. Blood cholesterol should be that should be low, right? So for our healthy life, and cholesterol synthesis enzyme also, that is inhibited by this statin. Okay, so these all are the bioactive molecules produced by the microbes. The next important one is microbes in sewage treatment. So how the microbes are going to help in the treatment of the sewage? Then first thing is, what is sewage, that you should know, right? So what is sewage means? Large quantities of wastewater are generated every day in cities and the towns. You know that the wastewater that is collected in cities and large towns, a major component of this waste is human excreta, and municipal wastewater is also called as sewage. Okay, the main constituent of this sewage water, that is the human excreta and some other chemicals, some other different plastics, so some disposable waste also present inside this sewage treatment. Okay, sewage, so municipal wastewater, so the what is collected from the households, that is also called as the sewage. Then it contains large amount of organic matter and microbes, many of them are pathogens. The microbes, they cause the diseases. Okay, and here some microbes are used to treat the sewage. Okay, before disposal, and sewage is treated in a sewage treatment plants, so to make it less polluting. So you know that the sewage water that is directly not released to the soil, before that that sewage water that will undergo some treatment, okay, to control the pollution, to remove the some harmful chemicals, harmful organic matters, so that are present in that water. So that is what the sewage treatment. Sewage water is done by heterotrophic microbes naturally present in the sewage. This treatment is carried out in the two stages. ಕಾಮನ್ ಆಗಿ ಈ ಸೀವೇಜ್ ಟ್ರೀಟ್ಮೆಂಟ್ ಅನ್ನ ಮಾಡಿದಾಗ ಏನಾಗುತ್ತೆ, ಅದರಲ್ಲಿರುವಂತಹ ಮೈಕ್ರೋಬ್ಸ್ ಇರ್ಬೋದು, ಕೆಲವೊಂದು ಆರ್ಗ್ಯಾನಿಕ್ ಕಂಟೆಂಟ್ಸ್ ಇರ್ಬೋದು, ಸೊ ವಿಚ್ ಇಸ್ which will not reach the soil, and the pollution will be less. Okay, so pollution kadme agutte, disease spread agodu kadme agutte. So the sewage treatment is important. So in this sewage treatment also some microbes are used. Okay, which are all the microbes used in this? Let us see. This sewage treatment that takes place in two steps. The primary treatment. Okay, so in primary treatment and the secondary treatment, two steps are there. In the primary treatment, these treatment steps basically involve physical removal of the particles large and small from the sewage through the filtration and the sedimentation. So illen ಮಾಡ್ತಾರೆ. So just imagine this is the sewage tank, okay, which collected the sewage, which contain the waste materials, human excreta, everything that is present in this sewage. So in the primary treatment, what happens? It will undergo the filtration. So this water, they transmit it to the filter. Some uh, floating waste like plastics, debris, and different physical waste materials are removed by this filtration. ಸೊ ಇಲ್ಲಿ ಫಿಲ್ಟ್ರೇಷನ್ ಮಾಡೋದ್ರಿಂದ ಏನಾಗತ್ತೆ, ಕೆಲವೊಂದು ಸಾಯಿಲ್ ಇರ್ಬೋದು, ಸ್ಮಾಲ್ ಸ್ಟೋನ್ಸ್ ಪೆಬಲ್ಸ್ ಇರಬಹುದು, ಹಾಗೆ ಕೆಲವೊಂದು ಪ್ಲಾಸ್ಟಿಕ್ ಐಟಮ್ಸ್ ಇರ್ಬೋದು. ಸೊ ಅದೆಲ್ಲ ಫಿಸಿಕಲಿ, so this is the primary treatment, means the first thing is physical removal should be there. After that, primary treatment. Then these are removed in stages. Initially, floating debris is removed by sequential filtration. Then grit, that is removed by the sedimentation. So first step ಅಲ್ಲಿ primary treatment ಅಲ್ಲಿ first step ಅಲ್ಲಿ ಏನ್ ಮಾಡ್ತಾರೆ, ಫ್ಲೋಟ್ ಆಗುವಂತಹ ಕೆಲವೊಂದು ಡೆಬ್ರಿಸ್ ಅನ್ನ ರಿಮೂವ್ ಮಾಡ್ತಾರೆ. ದೆನ್ ಇಟ್ ಈಸ್ ಅಲೌಡ್ ಟು ಸೆಟಲ್. ಓಕೆ, ಸೊ ಇಲ್ಲಿ ಸೆಡಿಮೆಂಟೇಶನ್ ಅಂದ್ರೆ ದ ಹೆವಿಯರ್ ಪಾರ್ಟಿಕಲ್ಸ್ ಲೈಕ್ ಸಾಯಿಲ್, ಸ್ಯಾಂಡ್, ಗ್ರಿಟ್ಸ್, ಸೊ ದೀಸ್ ಆಲ್ ಆರ್ they will they settle down to the base of this treatment tank, and this is removed. So that is called as the sedimentation. All solids that settle from the primary sludge and the supernatant forms the effluent. The effluent from the primary settling tank is taken for the secondary treatment. Okay, so this is called as the sediment, and upper one, the water is there, no, the sewage water is there, no, so that is called as the effluent. Okay, so or supernatant forms the effluent. Okay, so this is the supernatant where the remaining one looks like in the settled down in the form of sedimentation, and upper one, that is called as the supernatant, that is called as the effluent. Okay, and this effluent that is transfer for the secondary treatment. ಸೊ ಇಲ್ಲಿ ಎಫ್ಲುಯೆಂಟ್ ಮೇಲ್ಗಡೆ ಏನಿದೆ ಅಲ್ವಾ, ಸೆಡಿಮೆಂಟೇಶನ್ ಆದ್ಮೇಲೆ ಮೇಲ್ಗಡೆ ಇರುವಂತ ಸಿವೇಜ್ ವಾಟರ್ ಅನ್ನ ಸೆಕೆಂಡರಿ ಟ್ರೀಟ್ಮೆಂಟ್ಗೆ ಅದನ್ನ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡ್ತಾರೆ ಫಾರ್ ದ ನೆಕ್ಸ್ಟ್ ಒನ್. ಓಕೆ, ಲೆಟ್ ಅಸ್ ಸಿ ವಾಟ್ ಹ್ಯಾಪನ್ಸ್ ಇನ್ ದ ಸೆಕೆಂಡರಿ ಟ್ರೀಟ್ಮೆಂಟ್. ಸೊ ಇನ್ ದ ಸೆಕೆಂಡರಿ ಟ್ರೀಟ್ಮೆಂಟ್, ಸೊ ಪ್ರೈಮರಿ ಟ್ರೀಟ್ಮೆಂಟ್ ಅಲ್ಲಿ ಸೆಡಿಮೆಂಟೇಶನ್ ಆಗಿದೆ, ಹಾಗೆ ದ ರಿಮೈನಿಂಗ್ ಒನ್ ದಟ್ ಇಸ್ ಟ್ರಾನ್ಸ್ಫರ್ ಟು ದ secondary treatment. So here, or it is also called as biological treatment. The primary effluent is passed into the large aeration tanks where it is constantly agitated mechanically and air is pumped into it. So here, this effluent that is transferred to the secondary treatment where it get more air. Okay, so the aeration is provided with the pump artificially, or the more air is, that is called as the aeration. Okay, so here what happens, this allows the vigorous growth of useful aerobic microbes into flocks, means masses of bacteria associated with fungal filament to form the mesh like structure. So ಇಲ್ಲಿ ಆಕ್ಸಿಜನ್ ಅಬಂಡೆಂಟ್ ಆಗಿ ಸಿಗ್ತಾ ಇರೋದ್ರಿಂದ ಏನಾಗತ್ತೆ, ಇಲ್ಲಿ ಆಲ್ರೆಡಿ ಕೆಲವೊಂದು ಮೈಕ್ರೋಬ್ಸ್ ಪ್ರೆಸೆಂಟ್ ಇರುತ್ತೆ. ಓಕೆ, ದೇ ಗ್ರೋ ರ್ಯಾಪಿಡ್ಲಿ. ದ ಏರೋಬಿಕ್ ಮೀನ್ಸ್ ವಿಚ್ ಯೂಸ್ ದ ಆಕ್ಸಿಜನ್ ಆರ್ ವಿಚ್ ಯೂಸ್ ದ ಆಕ್ಸಿಜನ್ ಫಾರ್ ದರ್ activity. So aerobic bacteria, so they grow very fastly, and sometimes the bacteria, aerobic microbes, they forms the flocks. So what is flocks? That is the bacteria associated with the fungal filament to form the mesh like structures. Okay, so which form the mesh like structure also. Then while growing, so these microbes are growing inside this aerobic condition. They consume the major part of the organic matter in the effluent. So you know that some organic matter, some harmful organic compounds are present inside that water, that is consumed by the growing bacteria, growing organisms, okay, growing microbes. So the BOD refers, this significantly reduces the BOD, that is the biochemical oxygen demand. ಸೊ ಇಲ್ಲೇನಾಗತ್ತೆ ಅಂದ್ರೆ ಈ ಮೈಕ್ರೋಬ್ಸ್ ಇದ್ರೊಳಗಡೆ ಗ್ರೋತ್ ಆಗ್ತಾ ಇರತ್ತೆ ಅಲ್ವಾ, ಅದೇನ್ ಮಾಡತ್ತೆ ಅಂದ್ರೆ ಅದ್ರೊಳಗಿರುವಂತಹ ಆರ್ಗ್ಯಾನಿಕ್ ಮ್ಯಾಟರ್ಸ್ ಅನ್ನ, ಕೆಲವೊಂದು ಕೆಮಿಕಲ್ಸ್ ಅನ್ನ ಅದೇನ್ ಮಾಡುತ್ತೆ, ಕನ್ಸ್ಯೂಮ್ ಮಾಡುತ್ತೆ. ಸೊ ದಟ್ ಡಿಕ್ರೀಸಸ್ ದ ಬಯೋಕೆಮಿಕಲ್ ಆಕ್ಸಿಜನ್ ಡಿಮ್ಯಾಂಡ್, ದಟ್ ಈಸ್ BOD. BOD refers to the amount of oxygen that would be consumed if all the organic matter in 1 liter of ವಾಟರ್ ಆಕ್ಸಿಡೈಸ್ಡ್ ಬೈ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಸೊ ಇಲ್ಲಿ ಎಷ್ಟು ಆಕ್ಸಿಜನ್ ಬೇಕು ಈ ಒಂದು ಲೀಟರ್, ಒಂದು ಲೀಟರ್ ಅಲ್ಲಿರುವಂತಹ ಆರ್ಗ್ಯಾನಿಕ್ ಮ್ಯಾಟರ್ ಅನ್ನ ಕನ್ಸ್ಯೂಮ್ ಮಾಡಲಿಕ್ಕೆ ಹೌ ಮಚ್ ಆಕ್ಸಿಜನ್ ಇಸ್ ರಿಕ್ವೈರ್ಡ್, ದಟ್ ಈಸ್ ಕಾಲ್ಡ್ ಆಸ್ ದ BOD. ಓಕೆ, ಸೊ BOD ಇಸ್ ಹೈ ಮೀನ್ಸ್ ದ ವಾಟರ್ ಈಸ್ ಮೋರ್ ಕಂಟಾಮಿನೇಟೆಡ್. BOD ಜಾಸ್ತಿ ಇದೆ ಅಂದ್ರೆ ಅರ್ಥ ಏನು, ವಾಟರ್ ತುಂಬಾ ಕಂಟಾಮಿನೇಟ್ ಆಗಿದೆ ಅಂತ, ದ ವಾಟರ್ ಇಸ್ ಪೊಲ್ಯೂಟೆಡ್ ವಾಟರ್. ಓಕೆ, ಸೊ ಹಿಯರ್ BOD refers to the growing microorganisms. They use the some chemicals with the help of oxygen, and they reduce the BOD. Okay, then the sewage water is treated till the BOD is reduced. BOD ಕಡಿಮೆ ಆಗೋ ತನಕ ಅದೇನ್ ಮಾಡ್ತಾರೆ, so that is treated with the in that plant. The body BOD test measures the rate of uptake of oxygen by microorganisms in a sample of water. Thus, indirectly, BOD is measure of the organic matter present in the water. So, that BOD that refers to the organic matter present in that. Okay. The greater the BOD of wastewater, more its polluting potential. If the BOD is more, then the pollution is also, that water is also more polluted. So this is the diagrammatic representation where the primary and secondary treatment takes place. Yes, just see here, that is the raw sewage. So the raw sewage, that is filtered. Okay, so that is filtered by using the filters. Then it is transferred to the primary treatment, where the sediment that is also removed. Okay, sediment is also formed and effluent is also formed. Sedimented, where the settled down waste is removed. Then the effluent, which is the above one, water, sewage water, that is transferred to the secondary treatment. See here, secondary treatment in this tank. So continuously air is provided here. Aeration. Okay, so aeration means here what happens, the growth of the microbes takes place, and they consume the organic matter, and they consume the oxygen. They start to reduce the BOD, biological oxygen demand. Okay, when the BOD is less means the water is clear, when the BOD is more means the water is polluted. Okay, so these steps we have completed now. Okay, so then after the secondary treatment, next nodi, once the BOD of the sewage or wastewater is reduced significantly, the effluent is passed to the settling tank where the bacterial flocks are allowed to sediment. This sediment is called as activated sludge. Okay. So next ಏನ್ ಮಾಡ್ತಾರೆ ಅಂದ್ರೆ after the when the BOD is less, when the water become clear, then it is transferred to the next layer where the bacterial flocks means the bacteria and fungi, they form the um, what you call mesh like structure, so that is allowed to settle. Okay, so that will get settled and that is called as activated sludge. A small part of the activated sludge is pumped back to the aeration tank to the serve as the inoculum. ಈ activated sludge ಅಂದ್ರೆ ಅದರಲ್ಲಿ ಏನಿದೆ, so that contain the microbes. Okay, that microbes again transfer back to the aeration tank. So ಆ microbes ಇರುವಂತ ಸ್ವಲ್ಪವನ್ನ ಏನ್ ಮಾಡ್ತಾರೆ, ಅದನ್ನ ಏರಿಯೇಷನ್ ಟ್ಯಾಂಕಿಗೆ ಮತ್ತೆ ಟ್ರಾನ್ಸ್ಫರ್ ಮಾಡ್ತಾರೆ. That is called as the activated sludge and which act as the inoculum. Okay, the remaining major part of the sludge is pumped into large tanks called as anaerobic sludge digesters. So next, remaining part of that sludge, that is transferred to the anaerobic condition. So ಇಷ್ಟೊತ್ತೇನಾಯ್ತು aerobic condition ಅಲ್ಲಿ ಇತ್ತಲ್ವಾ. ನೆಕ್ಸ್ಟ್ the remaining one is transferred to the anaerobic condition where the oxygen is absent. So here some kinds of bacteria which grow anaerobically digest the bacteria and the fungi in the sludge. So they digest all the bacteria and fungi which is present in the sludge, and also they produce some gases like methane, hydrogen, sulfide, and the carbon dioxide. These gases from biogas and can be these gases form the biogas and can be used as a source of energy, and it is inflammable. And the effluent from the secondary treatment plant is generally released into the natural water bodies like rivers and streams. So ಇಲ್ಲಿ ಏನ್ ಮಾಡಿದ್ರು ನೋಡಿ, here the effluent from the after the aeration. So after aeration ಆದ್ಮೇಲೆ ಏನ್ ಮಾಡಿದ್ರು, that is transferred to the another tank where the bacterial flocks, they should settle down, they form the sediment. So ಈ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಮತ್ತೆ ಬ್ಯಾಕ್ಟೀರಿಯಲ್ ಫ್ಲಾಕ್ಸ್ ಅನ್ನ ಏನ್ ಮಾಡ್ತಾರೆ, again they transfer it to the aeration tank which act as the inoculum which helps to reduce the BOD of the next treatment. Okay, and remaining one, that is transferred to the, again in one more column should be there, that is transferred to the anaerobic condition treatment. Okay, so here another tank where the anaerobic condition is present. Okay, so in that what happens, the remaining bacteria and fungi, they will get digested by the anaerobic bacteria, and also they releases some gases that is inflammable. They release some biogases by digesting the remaining things in this, and they act as a they help to produce some biogases. Okay, after this secondary treatment, the sewage water, all the way or the wastewater, that is released to the water sources like a river and the pond like that. Okay, so that is very which will not cause the water pollution. So like this sewage treatment that helpful to remove the sewage different disease causing microbes, everything, so that is removed by the sewage treatment. Okay, here the programs like Ganga Action Plan and Yamuna Action Plan, so that was initiated by our Ministry of Environment and Forest of India. So what is this Ganga Action Plan and Yamuna Action Plan? So it started on the 14th January 1985 to improve the water quality of treatment of domestic waste. So sometime what they will do, the wastewater that is collected from the households, they directly leave into the river. Okay, but it causes the diseases to the organisms which is present in water and also human beings, that is not good one. So it also causes the water pollution. So Ganga action plan and Yamuna action plan, and different action plans were initiated. This was withdrawn on 31st March 2000. Then Ganga action plan 2, so that is analyzing the quality water quality of the river Ganga. Then Yamuna action plan. So Yamuna Action Plan, that was started in 1993. The Yamuna Action Plan was funded by a loan from the Japan Bank of International Cooperation of to the National River Conservation and Directorate Government of the India. So these all action plans, that is aimed to reducing the organic matter and thus reducing the BOD loads at a various locations in Yamuna. Okay, so these are the main goals of the these action plans. Then microbes in production of the biogas. So biogas, that is mainly that contains the methane, right? So this methane that is produced by the microbial activity. So here the type of gas produced depend upon the microbes and which organic substrate that utilized. So the example cited in relation to the fermentation of the dough, cheese and making production of beverages, the main gas produced was carbon dioxide. But certain bacteria, they grow anaerobically and on cellulose material they produce the methane gas, okay, which depend which grow anaerobically, and they digest the cellulose material and release the gas that is called as the methane. So the commonly the bacteria which helps to produce the methane gas, they are called as methanogens. Okay, so you can see the methanogens here, they are called as the methanogens. And one such common bacterium is methanobacterium. That methanobacterium, that is uh, that is helping to form the methanogen gas. Okay, so this bacteria commonly found in the uh, rumen of the cattle. Okay, these bacteria are also present in the rumen of the some cattle, and a lot of cellulosic material present in the food of cattle also present in the rumen. So here, yes, how the biogas production takes place here. Just see here. So dung and water that is mixed and that is reached to the anaerobic sludge digester. Okay, so also this cow dung that contains the anaerobic bacteria, methanogens and water. So in the anaerobic condition, they produces the gas, they digest the cellulosic material and they release the gas that is called as the methane gas. So this gas is collected in the particular gas holder. Okay, so if the gas holder is up means the more methane gas is produced. Okay, then which is connected with the gas pipe and that is connected to the household. So this is how the biogas is produced by using the um, by using the cow dung. The biogas plant consists of concrete tank. Okay, you can see the concrete tank. This is the concrete tank. So which where the waste collected with and slurry of dung is fed. A floating cover is placed over the slurry which keeps rising as the gas produced in a tank due to the microbial activity. So this is the gas holder. If the more gas is produced, that float on this digester. Okay, then the biogas plant has an outlet which is connected to the pipe to supply the biogas. So this is the outlet to supply the biogas to the household. and the spent slurry is removed through the another outlet and may be used as a fertilizer. So here the slurry that is reached here, no, that is collected with the another pipe, and that we can use as a fertilizer. Cattle dung is available in large quantities in rural areas where the cattle are used for variety of purpose. Okay, so this is how the biogas is produced. Next topic is microbes as a biocontrol agents. What is the meaning of biocontrol agent? Biocontrol agent that refers to the methods, the they use the some biological microbes or living organisms to control the disease and pest. Okay, living organ, not chemicals, living organisms only used to control the some diseases and some other pest causing disease, pest which cause the diseases. So in agriculture there is a method controlling of pest that lies on the natural predation rather than the introduction of the chemicals. Okay. So the very familiar beetle with red and black markings, so that is called as the ladybird. So this ladybird, that is the organism, and dragonflies are useful to get rid of the aphids and mosquitoes. These all are the some organisms which we use against the some used as a biocontrol agents. But here which microorganisms we should think. Okay, so here some microorganism that is called as bacillus thuringiensis bacteria. So bacillus thuringiensis, that is called as bt. Okay, which produces the toxin that is called as bt toxin, which the bacteria which produces the toxin that is called as bacillus bt toxin, that is against the some caterpillars. Okay, which control the caterpillars. So that is the one of the biocontrol agent, and these are available in sachets, dried spores which are mixed with the water and sprayed onto the vulnerable plants. So that is help to control the this caterpillars. In the gut of the larvae, the toxin is released and larvae get killed. When this Bacillus thuringiensis that enter to the larvae body, that is caterpillar, then that will that toxin that kills the caterpillars. Okay, so this was used in the genetic engineer. Genetic engineering ಏನ್ ಮಾಡಿದ್ದಾರೆ, ಈ BT toxin you produce ಮಾಡುವಂತ Bacillus thuringiensis gene ಅನ್ನ, so they introduced in the plant, and the plants are now naturally producing the BT toxin against these larvae. And one more is biocontrol agent is trichoderma. Trichoderma species are free living fungi that are very common in the root ecosystem. So trichoderma that is present with the root, and they are effective biocontrol agents of several pathogens. So ಅದನ್ನ biocontrol agent ಆಗಿ ಯೂಸ್ ಮಾಡ್ತಾರೆ. That is the trichoderma fungi. Baculovirus are pathogens that attack the insects and other arthropods. So this baculovirus that is also used as a biocontrol agent against the some arthropoda and some pathogen, and trichoderma also. The majority of baculovirus used the biocontrol agents are in the genus nucleopolyhedrovirus. So this is the excellent for the excellent biocontrol agent. The one more thing, trichoderma, then baculovirus and bacillus thuringiensis, these all are example for the microbes as a biocontrol agent. And microbes as a biofertilizer. Biofertilizers means they are producing, they are helping for the growth of the plants, they are providing the nutrients. Okay, here, the main source of biofertilizer, that is bacteria and fungi and the cyanobacteria. Here, There's some bacteria, they are present in the nodules of the leguminous plants like pea plant and groundnut. So these help for the absorption of the nutrients and symbiotic association of the rhizobium. So rhizobium, so that is the bacteria that is present in the roots of the leguminous plants that fix the atmospheric nitrogen. Atmospheric nitrogen ಅಂದ್ರೆ ವಾತಾವರಣದಲ್ಲಿರುವಂತ ನೈಟ್ರೋಜನ್ ಅನ್ನ ಏನ್ ಮಾಡುತ್ತೆ, ಪ್ಲಾಂಟ್ಸ್ ಗೆ ಅವೈಲೇಬಲ್ ಆಗೋ ತರ ಅದ್ವೇನ್ ಮಾಡುತ್ತೆ, ಕನ್ವರ್ಟ್ ಮಾಡುತ್ತೆ. ಓಕೆ, that is the first one. And other bacteria can fix the atmospheric nitrogen are, so that are free living in soil like azotobacter and azospirillum. So ಇನ್ನು ಕೆಲವೊಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಏನ್ ಮಾಡುತ್ತೆ, ಅಟ್ಮಾಸ್ಫಿಯರಿಕ್ ನೈಟ್ರೋಜನ್ ನ ಕನ್ವರ್ಟ್ ಮಾಡಿ ಪ್ಲಾಂಟ್ಸ್ ಗೆ ಅವೈಲೇಬಲ್ ಆಗೋ ತರ ಮಾಡುತ್ತೆ, ದಟ್ ಈಸ್ ಅಜೋಸ್ಪಿರಿಲಮ್ and the azotobacter. But they are free living, they are not associated with the roots of the plant. ಬಟ್ ರೈಜೋಬಿಯಂ ಅನ್ನುವಂತ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಏನಾಗಿದೆ, ದಟ್ ಈಸ್ ಅಸೋಸಿಯೇಟೆಡ್ ವಿತ್ ದ ರೂಟ್ಸ್ ಆಫ್ ದ ಲೆಗ್ಯುಮಿನಸ್ ಪ್ಲಾಂಟ್ಸ್. Then fungi are known to form the symbiotic association with plants, that is called as mycorrhiza, and many members of genus Glomus form the mycorrhiza. So ಇಲ್ಲಿ fungi ಏನ್ ಮಾಡುತ್ತೆ, fungi they are associated with the plant's root, that is called as mycorrhiza, and they also producing the nutrients, they also help to absorb the nutrients for the plants. Okay, the fungal symbiont in this association absorb the phosphorus from the soil, especially phosphorus nutrient. Okay, plants having such association show the other benefits such as resistance to the root-borne pathogens, which control the root-borne pathogens also, and tolerance to the salinity, drought, and overall growth and development. So ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ ಮೇನ್ ಆಗಿ ಪ್ಲಾಂಟ್ ಗ್ರೋತಿಗೆ ಏನ್ ಬೇಕು, ಫಾಸ್ಫರಸ್ ಮತ್ತೆ ನೈಟ್ರೋಜನ್ ಬೇಕು. ನೈಟ್ರೋಜನ್ ಡೈರೆಕ್ಟ್ಲಿ ಅವೈಲೇಬಲ್ ಇಲ್ಲ, ದಟ್ ಶುಡ್ ಬಿ ಕನ್ವರ್ಟ್. ಓಕೆ, ಫಾರ್ ದಿ atmospheric fixation means converting the nitrogen into the simple organic forms, simple forms, that is called as the nitrogen fixation. So for that rhizobium and azospirillum, azotobacter are helping, and fungi that is help to absorb the phosphorus and also control the root-borne pathogens and also help for the overall plant development. Then one more microbe, that is the cyanobacteria. This cyanobacteria, that is the autotrophic microbe, and which also fix the atmospheric nitrogen. Nitrogen fixation ಅಂದ್ರೆ ನಿಮ್ಗೆ ಗೊತ್ತಿದೆ, ವಾತಾವರಣದಲ್ಲಿ 78% ನೈಟ್ರೋಜನ್ ಇದ್ರೂ ಕೂಡ ಏನಾಗೋದಿಲ್ಲ, ದ ಪ್ಲಾಂಟ್ಸ್ ಆರ್ ನಾಟ್ ಏಬಲ್ ಟು ಯೂಸ್ ದಟ್ ಡೈರೆಕ್ಟ್ಲಿ. ಸೊ ದಟ್ ಶುಡ್ ಬಿ ಕನ್ವರ್ಟ್, ದಟ್ ಕನ್ವರ್ಷನ್ ದಟ್ ಈಸ್ done by the some cyanobacteria, some other rhizobium bacteria. Okay, so here Annabena, nostoc, so these all are, they fix the atmospheric nitrogen. And blue green algae, that is the cyanobacteria, also add the organic matters to the soil also. Okay, so this is how the microbes that are helping as a biofertilizers, microbes used as a biofertilizers. Okay, means biofertilizers means the living organisms which provide the nutrition to the plants, and biocontrol agents means the living organisms which are used to control the pathogens as a without using the chemicals, that is the biocontrol agents. So many bio microbes we have seen, that is bacillus thuringiensis, then trichoderma, baculovirus, these all are the example for that. Okay, so I hope this concept is clear. Now in this chapter what we studied, so microbes in a human welfare. So what are all the points we studied here? The first things, microbe in households, and microbes in the production of beverages, then antibiotics, then enzymes, and organic acids, and some organic acids and bioactive molecules also. These all we have seen in the industrial uses. Then sewage treatment, after that biogas production, and also biocontrol agent, and the microbes as a biocontrol agent and the microbes used in biofertilizers. So these all things that we have covered in this video. I hope these all things that is very helpful to face the board exam. Thank you. Bye bye.